about those 4 biomolecule groups. When we talk about biomolecules, of course, those are molecules needed for life. The fact that we're calling them molecules means they're held together with covalent bonds. We know that they are also organic, correct? Meaning what? They all contain carbon. And we're going to go through and talk about the 4 groups of biomolecules, which you learned about in Bio 1, studied for your pop quiz. By the way, 6th period was glad, very proud that they took away the overall championship from 7th period on that quiz. Those rascals, right? What are those 4 biomolecule groups, guys? Carbohydrates. What's another one?
Lipids.
Lipids. What's another one? Proteins. And lastly, nucleic acids. So those 4 biomolecule groups: carbohydrates, lipids, proteins, and nucleic acids. Okay? We're going to be looking at specific information about each of those, including what atoms make up those, are common to those groups. That's something we want to keep track of. So first of all, I happen to have a biomolecule here. How handy is that? Yeah? That's handy, right?
Mm-hmm.
So when we look at these biomolecules, for the most part, although not for all of them, as we'll talk about, most of them are large molecules made up of many repeating units, okay? And I think you remember this vocabulary from Bio 1. What is that word that means many units that would describe these biomolecules? Polymers. They're polymers. Poly means many, and mer means unit. Okay, so they're polymers made up of many units, and what we're going to be doing, and of course you did in Bio 1 as well, is identifying those individual building blocks that make up these polymers, those individual building blocks being called monomers, which means single unit or 1 unit, right? So most of them are polymers. We can clearly identify those individual building blocks that make them up. And so the question is, how do we join together all of those monomers to make these large polymers, to make these large biomolecules? Well, the reaction that is utilized to build these biomolecules is called dehydration synthesis. And it actually goes by 2 different names, but I like that name because it's descriptive when you are a root expert such as yourself. Yes? Because what does DE mean? What is that root? Yeah, remove away, right? Hydra? Water. So we are removing water, and synthesis means to build, to make. Remove or away water to make. That's what's happening in dehydration synthesis. What we're doing is joining together those monomers, and that occurs through the removal of a water molecule, okay? Now, there is another term that's used to describe this reaction that you need to know because you see it. It's also called condensation. So condensation and dehydration synthesis are synonyms. They refer to the same reaction. Now, we've already said that we are building biomolecules. So what type of bond is being formed as we join together these monomers? Covalent. So we are forming strong covalent bonds by removing a molecule of water in order to build these polymers. And we've already said, of course, that water is lost as a waste product. As you might expect, if we are building, it means that we have to put energy into the reaction. Okay, what's that word? By the way, we're going to be talking about it in a couple of units, but remember what energy in is? And is it endergonic or exergonic? Endergonic, right? It's an endergonic reaction. We have to put energy into it. And as all chemical reactions virtually require, it does require the action of enzymes. Now, question for you. Looking at this graphic of how dehydration synthesis occurs, knowing that at least one of those molecules has to be able to donate an OH in order to generate that water molecule, what functional group is going to be involved in dehydration synthesis? Hydroxyl, what's the other possibility? Carboxyl, right? So this is going to be a reaction if dehydration synthesis is occurring in which we either have a hydroxyl or a carboxyl group because we have to get that OH from somewhere, yeah? Okay, all right? Very good. Any questions on dehydration synthesis? So it should make sense to you. If I am building by removing water, how do you suppose I break? How do I split them apart?
Add water.
I've got to add that water back, okay? And again, the name is descriptive. It's not anything you need to memorize. What do we call those reactions? hydrolysis or hydrolytic reactions. Hydro again means water and lys means to break, water to break. Okay, that's what we're doing in order to break these apart. Yes?
I don't get how moving water from something builds it.
It just allows for the formation of that covalent bond between the atoms that are left, the carbons, okay? Through the action of enzymes to hold it together. All right, so with hydrolysis, again, by adding that water back, we allow for that molecule to be broken apart, again, under the guidance with the catalysis of enzymes involved in that. And this is one of the many reasons that water is such an essential component. As a matter of fact, we talked about the fact when we grew those seedlings, I kept telling you they need a lot of water. Okay, seeds have to, what we call imbibe. They have to be able to take up water in order to germinate. In large part, that's to allow for these hydrolytic reactions to occur. Those seeds had stored glucose in them. What do we call stored glucose in plants?
Starch.
Starch, okay? That seeds, that little baby plant's gotta be able to break down that starch to glucose in order to start metabolizing, in order to start making ATP. And that's part of the purpose of that water, is to allow for the hydrolysis of starch so that that little baby plant, little baby seedling can start using it for energy. Okay. Any questions on that? Obviously, building requires energy. We know that energy is neither created nor destroyed, right?
Mm-hmm.
So what does breaking do? Releases energy, okay? It's gonna release energy, all right? Any questions on that? Alrighty, so we are going to start our discussion with proteins. Now, I'm sure in Bio 1 you started your discussion of biomolecules with carbohydrates. Why do we start with proteins? Well, there are two reasons for that. One, it has by far the most information associated with it, and I always feel like I'd rather front-end that information than do it the last couple of days before you're taking the exam. Secondly, you have to understand proteins to understand the AP lab that we're doing on Wednesday. So we're going to start with proteins and cover those first. They are, as I said, by far the most complicated of the groups that we're covering. So how important are proteins? Very. I mean, we can't overstate their importance. We associate proteins with the genetic code. Why so? What do proteins have to do with DNA? That's how DNA's instructions are carried out, right? That's what makes me, me, and you, is what our DNA tells our cells to do with regard to the making of proteins. When you look at each other, you are looking at proteins, right? Our hair, our skin, our nails, even the sclera of our eye, right? All of that made up of proteins. So they are critically important. We'll come back and look at some of their functions later. What we want to do today is talk about the structure of proteins. Monomers of proteins are amino acids. And looking at the elemental composition of these groups, which you need to know, what do you remember from Bio 1? What elements found in all proteins? CHO and CHON, right? Carbon, hydrogen, oxygen, and nitrogen found in all proteins. So in looking at amino acids, first of all, they have a basic structure. This is kind of like functional groups, right? You just need to learn it because you're going to see it all year long. So what is the basic structure of an amino acid? Well, it's an organic molecule. And so when we think of the word organic, we think of carbon. So we start with a carbon. That carbon goes by two specific names. We describe that carbon as the alpha carbon. We also describe that carbon as an asymmetric carbon. What do we mean when we call something asymmetric, or we refer to a carbon as being asymmetric? That's an organic term that you might as well become familiar with. What it means is that there are 4 different things bonded to that carbon, okay? So there's no symmetry there, there's no likenesses. Okay, so asymmetric carbon, arm down please.
So that's the same as the alpha carbon?
In this particular case it is, okay? All alpha carbons are asymmetric carbons, not all asymmetric carbons are alpha carbons. Got that? Asymmetric carbon is a more general term. It describes any carbon that has 4 different things bonded to it, and alpha carbon is used specifically to describe the carbon in amino acids.
Okay.
Okay? All right, so we got that alpha carbon, okay? The rest of this is descriptive. If you know your functional groups, you should never have a question about the structure of an amino acid. Its name is descriptive. What's an amino group look like? You got a nitrogen along with two hydrogens. And by the way, guys, I only changed colors because that pen wasn't working well. So it doesn't mean anything significant. So we've got this amino group bonded to my alpha carbon, my asymmetric carbon, and it is an organic acid. All organic acids have what functional group?
Carboxyl.
Carboxyl, correct? What does a carboxyl group look like?
C bonded to an
This is where it can
O.
get confusing, right? Do I put the double bonded oxygen in the OH here? No, because what does my carboxyl group include always? The carbon, right? So I've got a carbon double bonded to an oxygen with an OH. Everybody agree? How many different amino acids are there in life? 20. There are 20 different amino acids, okay? What makes each of those 20 amino acids different from one to another is what we call the R group. R stands for rest of the molecule. Each one of those 20 amino acids has a different R group. And of course, as we've discussed, the valence of carbon is 4. And so in order to fill the valence, we have a hydrogen, okay? Now, hear what I'm saying. You do not need to memorize the 20 different R groups. You do not need to know what the R group of cysteine is compared to lysine, compared to proline, compared to methionine. Okay, you don't need to know that. However, if I give you an R group, you should be able to derive some information from that. Yes?
Whenever they're drawing out the structures, are they cumulative? Like if you're in a carboxyl group, if you have to draw it, do we have to include like the amino group too? So in addition to the COOH, we put the.
If you're drawing an amino acid, you do, yes.